Thank you, Lisa. We meet again. And in the past, we've gone through a non-scientific introduction to the power of more comprehensive nutrition, as well as a scientific one. And I've tried to develop what I think is important, that you need to know that this information is vital to you to improve quality of health. And I call it personal, preventive, and interventional health management, which supports engineering and design of life that is coded in the genes. And it's far more economical than any other approach to health management, which is important because the United States has the highest healthcare costs of all nations in the world, and rates by the United Nations World Health Organization number 35th in its last survey of quality of life of citizens. In our last session, we talked about Pettinger's cats and how about 40 to 50% of what we eat being raw gives your body the molecules as they're made in nature and not modified by the heat of cooking. And that's important. And there's one thing I didn't go into because there was so much additional information at the end trying to cover what people might be asking, is that if you remember that in the type B cats that in their groups, that everything they ate was cooked and that their health had deteriorated. And after the experiments were done, they had a running area outside their shelter that the cats that had raw meat, the weeds were vigorous and grew greatly, but very little vegetation grew in the area where the urine and feces were on the ground from the totally cooked diet cats. Well, I think that's important because during the Korean War, the military bases contracted with local individuals that had a business to go around in the communities and collect excrement from the population of Koreans, and that's a fancy word for saying urine and feces, and used it as fertilizer on their farms. Well, they made contracts with the military to come in and empty once or twice a week the latrines of the military and treated in the same manner. And they began to complain that the material they took from the latrines of the American military was not as good on the farm as a fertilizer of that of the diet of the local Koreans, which was more natural. And it's, as I told you in the DVD, that it was kind of chilling to me at the end of presenting what I showed you. They said that the Society of the United States is following down the same road as the diet of the totally cooked cats, and that we were having new diseases that had never been seen before. And I think it's kind of a replay of the history of discovering vitamins, that there are many other additional nutrient molecules that, for example, by choice or by cooking, you destroy. And the very fact that the excrement from the military of our American military was not as fertilizing, much like the A and B different groups of cats, is significant. Now, in this session, which we are going to go to, it's going to be about wound healing and burns and the history of and current science that supports past anecdotal folk use of the aloe plant. So with that, we will go to the PowerPoint presentation. This lecture topic will be the use of the aloe leaf gel in history for wounds and burns. And we'll talk about the benefits. And what I want to get across is that modern science is documenting ancient use. Now, it can be found in ancient history, that's even before our calendar started, that aloe vera leaf gel was applied to wounds for healing, and it's oldest use that can be documented. And significant in history that under Alexander the Great of Macedonia, which is when the Greek Empire dominated the Mediterranean region, the old Persian Empire, and extended into northern India, it is written that Alexander went to the island of Sakarta in the Red Sea because it was rumored they had the best aloe plants that had the best wound healing potential. And Alexander the Great had a war train, which in warfare was one of the first things about recognizing you had to have supply of food and munitions and everything to support his army. They had wagons that had growing plants that they could break the leaves off, because the wounds of that time were primarily due to swords, to knives, to spears, to arrows, and the hand-to-hand combat injuries. This, I think, is significant in that I had the personal experiences, but as a youth on a blackland dryland farm in Texas, my mother kept an aloe plant in the kitchen, in which we used it to break off the leaf and smear the leaf gel for chigger bites, mosquitoes, any insect bite. It was good for sunburns and for thermal burns and for minor skin blisters and cuts. And the reason we liked it is because it relieved the pain and the itching. And incidentally, by retrospect, no infections occurred. Now, for a manner review, I'd like to point out that in the research paid for by Carrington Laboratories, it raised $20 million to try to make a new pharmaceutical drug from whatever was in the aloe leaf gel that gave those properties that we were talking about, and that academic institutions found out that the active ingredient was acetylated mannose, less than 1% of the solids in the gel, which is primarily water and leaf gel aloe, and that this acetylated mannose, it's also called Acemannan, was used in cellular synthesis. Very important to say use, because it became part of the structure in the second step of cellular synthesis inside the cells of the body. All the cells initially have a little assembly line, which is in this presentation, which is a cartoon, because electron microscopy of this is challenging to interpret. And this comes from not our research or that of Carrington, but from an article in Annual Reviews of Biochemistry 1985 by the biochemist Kornfeld, husband and wife team from Washington University at St. Louis. And at the end of it, it's a review paper. And at the end of it, there's citations of research in the previous 20 years from at least 300 different published articles that eventually defined this assembly line, that they put it all together. And significance that I found this about 11 o'clock in the medical school library, this article. And we're working with a graphic artist. This cartoon is not in the paper, but it describes how in the center you see the biochemistry of what happens in the second step there, where you see number 2. The first step is the ribosome. And the second step, sugars are added to chains of amino acids that are called peptides. And in the endoplasmic reticulum, 9 molecules of the active ingredient from the aloe plant, the acemannan, are organized in 3 chains, and they're on the surface membrane of all the cells in the body. And this domain has a function for cell-to-cell communication and instructions throughout all the cells to carry out harmony among all the cells. Now, this article focused on the mannose, that was the principal material or molecule we found in aloe leaf, but it is if you step back and really look at it, all the different amino acids, fatty acids, sugars, vitamins, minerals, and phytonutrients from plant kingdom are put together on this assembly line, depending on which gene of the 30,000 genes is activated to make a bioactive structure function molecule. And so we're going to explore how this acemannan works, and the proof that it worked that comes from ancient observations, the modern biochemistry of how on this assembly line that the healing process of the steps that take place to heal a wound or a burn actually occurs. So with this, we'll go to the second slide after this thing. Now, in ancient history, in many primitive lands and nations, their local language for the aloe plant, and there are over 200 species in nature, could be translated into English that essentially meant the doctor or the medicine plant. It's been recognized in primitive civilizations about its power to support good health. It was use of aloe as in Egyptian formulations, in India and the Ayurveda medicine of India, and in Oriental medicine, aloe was utilized as an ingredient in many herbal natural mixtures. Of significance, I believe, that aloe is mentioned both in the Old Testament and the New Testament, meaning the ancient Hebrew writings, and then the more current writings since the birth of the young carpenter from Galilee. And it's been used in incense and in scents and perfumes. And it's mentioned in Numbers 6 that the aloes were planted by God for the people beside the river in Judea. In Psalms 45, it speaks of the garments are sprinkled with myrrh and aloes of the bed of the loved one, along with myrrh. In the Psalms of Solomon, it states, quote, "My bride, the bed is sprinkled with myrrh, frankincense, and aloes, and many spices." And then in John 19, Nicodemus embalmed Jesus of Nazareth with myrrh and aloes that were applied to his body. Thus, the value of aloe leaf gel is over 5,000 years in the Hebrew scriptures, and over 2,000 years in the New Testament. When we filed our patents for Carrington Laboratory, there were about 50 or 60 that I'll mention later. A academic person from Texas A&M brought me a translation from the second AD in Rome that Pliny the Elder wrote a essay on the range of health benefits of aloe. And this included about 30 uses that we submitted for the patent for Carrington Laboratories. And this included not only topical use of aloe, but also drinking it. And the benefits, particularly in the GI tract, are mentioned in this, as we had in our patents. And I mentioned this to point out that I'm just for you a messenger and servant, that I really haven't come up with anything new or invented anything. I'm just carrying information. It's important for you to know about this because the function of the brain is the mind, and the mind gives control to everything in the body, not only the physical systems, but the emotions and mental activity. The mind is a superior activity of the body. So what you know and think is important. And in the past, before this Pliny the Elder and our research in the modern era, the use of aloe was primarily for topical applications. Now, at Carrington Laboratory, brief review of how this got started in 1985 when I was asked to come and be a medical consultant. The same man that formed Carrington Laboratories had a small network marketing company called Avocare that had 12 aloe leaf gel products. There were lotions, potions, creams, and salves, but they also had a 32-ounce bottle, Carolo, it was called, a aloe leaf gel drink. And he came to my office at Dallas-Fort Worth Medical Center in August 1985 with a story that in 84, 85, he had received notes and faxes from 7 different states. Very important, these people did not know each other, that they were all AIDS patients that had been on their beds waiting to die of this new disease of unknown cause and no treatment. And they stated in these messages to him, as president of Avocare, that they drank his Carolo, and now they were back at work or in school with very few symptoms, if any, and that he thought that the company would want to know this. And what they were drinking was the stabilized Carolo beverage. In response to this, Carrington Laboratories was founded, and through individual personal fundraising, over $20 million were gathered to spend to develop a new FDA-approved antiviral drug against AIDS. And so working with Carrington, it was set up to do everything the FDA, or the Food and Drug Administration, requires for a new pharmaceutical drug, and it's quite a process. And due to the history of being used for skin problems, the first product developed was a wound gel, and it's called Carrington Wound Dressing. And I will tell you a little about that later. And then the second thing was with the U.S. Department of Agriculture, injectable aloe immunostimulant for animals was present, which had incredible benefits in animals. And before we're through, you'll see that a lot of things in the biochemistry of animals, if it works there, you can be pretty sure that it will work in humans too. One of the first things that happened is the alcohol concentration of the aloe leaf gel solids, which was called Manipole, was sent to the Parkland Burn Center, and Dr. Charles Baxter, who I had known as a student, reported that they took tissue from circumcisions of newborn babies that were put in tissue cultures, and they added to the culture media radioactive thymidine. And thymidine is one of the nucleic acids in the gene code, which is made up of 4 important molecules called adenine, guanine, cytosine, and thymidine, which is radioactive. And in these cultures, new fibroblasts grew out from this tissue from the head of the penis, and they could, with their instrumentation, show that the fibroblasts growing out of the tissue had a 400% increase in isotope. labeling. This was a documentation of accelerated cell growth. And this led to this mannopole, or this alcohol extract, being sent to Baylor School of Dentistry across town. And with oral ulcers, which were called aphus ulcers, are thought to be related to autoimmune diseases, they reported to about a 20 to 30% faster healing of these ulcers. Down at Texas A&M School of Veterinary Medicine, which was one of the 12 institutions, academic institutions, that were working on contract to fulfill what the FDA required in the way of science, they took miniature pigs. And the reason is that the skin of pigs responds more like the skin of human beings than other animals. And they were able to show that by topical application of the mannopole to experimental lacerations, in other words, their length and the depth of them was identical over and over, experimental wounds, that they healed 20 to 30% faster, and they had a stronger fibrous scar, but also less scarring. It was a stronger, less apparent scar. And one of the things they eventually noted, that the pigs they would take for their controls, which they had had experience with, how long it would take without any application, that even the control on the opposite side of the pig, where they would make the same kind of wounds, that they would heal faster. And that, in other words, that the blood circulating through one side picked up the active ingredient that was being applied to the experimental wounds, and it influenced the control wounds on the other side, and it accelerated their healing. It was carried by the blood. In addition, at this institution, they showed that rabbit eye wounds model heal faster with less scarring, which in the eye, that was extremely important. This work at Parkland had residents there, and one of the residents was very skeptical about the aloe, this natural non-drug approach, was causing this increased healing, and he thought it was the preservatives that were put into the wound gel or other addition things that were doing it. So he returned to his homeland. I think it was Costa Rica, I'm not sure, if I recall. And he did some experiments I'm going to show you to try to disprove that aloe was a benefit. And this is a paper that he that was published at Baylor in the journal Wounds of 1994. The title was "Evaluation of Acemannan, the active ingredient in aloe, and treatment of recurrent aphus stomatitis." And they, if you'll notice in this abstract, that they estimated, compared to what they've been doing in the past, that these ulcers healed about 1 day sooner. That and wound healing is really not all that significant. However, the discomfort of burning and pain was reduced by applying this to these ulcers. The resident from Parkland Burn Center went home, and this is what he did. He took a guinea pigs with a shaved area on their back, and this is a contraption that he set up to make sure he had a constant type of burn on the back of these research animals. And for 6 seconds at 250 degrees centigrade, he put a experimental burn on the back of these guinea pigs, 40 guinea pigs. This is what he found out, that if he used the typical FDA approved silverdene ointment to the burns of the guinea pigs. It took 45 days for the burns to heal. If you use the aloe gel, it took 31 days instead of 41 days. And he took the preservatives, the additives that were put with the aloe, and lo and behold, what he thought was the cause for healing was about 46 days. It was even slower by at least 1 day in these guinea pigs than the FDA approved ointment. Is another control. They just used plain gauze and over the burns, and it took 49 days. So in addition to this, it should take a notice in your mind, because we're going to show you that at a military burn center, that in humans, there was reduced pain, more rapid healing, scarring was reduced, and infections were avoided. And I want to point out that in this model of using an animal, that the information obtained was repeated again when you had a chance to expand on to animals. Now, at the University of Auburn, as we point out, this is an independent academic, it's a non-company conducted investigation. They just did it out on their own by hearing about the benefits of this aloe compound. And the Carrington Laboratories had nothing to do with it. And I mention this because so many critics will say, oh, these people were just playing to the financial source of the company and giving them good results. Well, this at Auburn, they got no financial benefit from what they reported. And that on the wounds of the pads that were put on dogs' feet, they said that the wounds healed 7 days sooner than if they used the triple antibiotics that is a gel that is typically used in veterinary medicine for wounds of dog pads. This was presented in the Journal of American Animal Hospital Association, and it got the volumes. It was 1992. It was also presented at a wound meeting society in August 93 in Amsterdam, Netherlands. So this is just independent, non-company information. Now, here is another paper trying to figure out why this works. And this was accelerated wound healing induced by macrophage stimulation in rats. This is basic science study, and that this is genetically controlled activity, a mechanism of action. And these researchers are graduate student and professors at Texas A&M School of Veterinary Medicine. But this was paid for by Carrington Laboratories. And some people will try to find fault with this because it was sponsored by the company that made the gel, but you know, they're putting their reputations on the line by publishing this in the Journal of Wound Repair and Regeneration. And again, I want to emphasize that macrophages are a special white cell in the peripheral blood that has many functions, and that this was a basic science experiment showing that this macrophage is active in this improved rate of wound healing. Now, here is a very important study that, again, is an independent wound center in Denver, Colorado. You see the authors there, and no one from Texas or Carrington Laboratories are involved. And certainly I wasn't involved. The title was a controlled randomized study of amorphous hydrogel, meaning this aloe wound gel, in diabetic ulcers, because diabetes too is virtually a silent epidemic in the developed countries. It was very serious in the United States, and they're plagued with non-healing wounds on their legs, feet, and toes. And oftentimes toes or their part of their foot is amputated because, and even on up the leg, because these ulcers won't heal. Well, what they found out is that with this active ingredient from the aloe that contains the acemannan, that there was improved healing, and they used as controls the type of management they used in the past, and the Carrington wound dressing improved the healing. This was presented in European and a Tissue Repair Society in 1994. And the important thing is that there was reduced cost, more effective healing as compared to the standard way of managing wounds of the diabetics. Now, as humans age and also as rats age, healing becomes slower. And this was a research project conducted at Texas A&M School of Veterinary Medicine, whereas they took old rats that they knew as controls, how long it took incited wounds on to heal, versus young rats. And they, by using the acemannan on applications, the old rats heal just like the young rats. And this was presented to Keystone Symposium Wound Repair in Colorado in 1993. Again, modern science validating what Alexander the Great had said before our calendar started. Here is another study that was conducted at Texas A&M School of Veterinary Medicine. They took the rate of healing of young, old, and even obese and even starved rats. In other words, poor nutrition. And even with poor nutrition and low caloric, and to the point that their muscle mass was decreased, as well as the old obese rats, that by using the acemannan gel topically on the wounds, they healed faster. And this was presented to the European Wound Repair and Healing Society in '93. Again, independent studies. I really found this paper of significance. The material submitted to MD Anderson Basic Science Radiation Experimental Department. You can see these authors here. And yes, this was material provided by the company. But again, these researchers at MD Anderson, which is an internationally recognized research center, that they took rats and shaved them and backs, and they found out they could give 9 gray, which used to be thought of as Roentgens, but I believe it's 100 Roentgens equal a gray, that they could take 9 more, 8 to 9 more gray radiation to the skin before they induced skin reactions or erythema in these rodents. Now, this was presented at Biophysiology in 1995, but I was present when the investigator, Elizabeth Travis, presented an oral presentation to the principals at Carrington Laboratory about this research project. And I will never forget, she stated that radiation can be administered sufficient enough to destroy any cancer cell. But she said the problem is that the radiation kills normal cells, and to give enough radiation to destroy any cancer, you'll kill the patient. And she pointed out that this topical gel, which allowed them to give 8 to 9 more gray radiation without inducing any erythema in the normal cells of the skin, that if you gave this orally or internally or by injection, and also provided it on the topical, that this protects normal cells from the damage from the radiation, which creates free radicals as its killing method. And the Acemannan quenches or neutralizes free radicals in normal cells, but doesn't do it in cancer cells, so the normal cells are protected. I thought this was very exciting, and that more money should be put in to follow up on this statement by Dr. Travis. However, the principals in Carrington Carrington Laboratories were focused on, they just had money to try to develop a new antiviral drug against AIDS, and that's where they're going to spend their money. So this was not pursued, and it's a project that very definitely needs to be followed up on, but you have to have research funds to be able to conduct any kind of research, but this is something that really needs to be investigated further. Well, as a consultant to Carrington Laboratories, all the material was put together to file patents for the company to protect its intellectual properties. And the title of the patent was Administration of Acemannan. And you will note that the chief medical officer for Carrington was Dr. Bill McAnally, and that research veterinarian Robert Carpenter from Texas A&M was on it, and I was listed as a co-inventor, and we assigned it to Carrington Laboratories, and the patent number is there, and it's issued 1992, and this patent is expired. But in the small print over there, the claims that we filed were to modulate, not stimulate or suppress, but modulate the immune system. And what that means, if the immune system or host defense is not strong, that it upregulates defense and healing. And if it's like in autoimmune diseases and overactive immune system and autoimmune diseases and say asthma, it suppresses the overactive immune system and brings it into a normal zone or homeostasis, balance, which the genes are programmed to establish. And of course, number 2 was enhanced wound healing, and it was found to be a systemic vaccine adjuvant. Vaccines make higher levels of antibody to protect the animal. That not only was there antiviral activity against the AIDS virus, but every animal virus and also a panel of human viruses, it prevented infection if it was on board in the patient or in a tissue culture. If you already allowed the animal or the tissue culture to be infected and the viral infection going, it immediately stopped the antiviral activity, and that in tissue cultures, it showed a rapid removal of viral proteins from the cells. So that's incredible antiviral activity. And the mechanism was looked into and further, and what it was doing was enhancing the general host protection against infections. And so it was found at institutions like at Texas Tech School, Medical School, that bacteria and fungus and other pathogens, and ultimately from orphanages in Africa, that malaria and chronic infection in children was arrested. So the very important thing is it does not poison the infection agent. What it does is boosts the body's ability to handle a foreign infectious agent. And so drug resistance, which is a big problem in using drugs, is not involved in this. And in fact, in tuberculosis, that was no drug would touch it. Tuberculosis was cleared because the body's defenses were increased by working through the genes rather than against the infectious agents. So that's very important to know. Also, number 5, that as you noticed with MD Anderson, that we found, particularly in animals, feline leukemia, that it was eliminated, and various animal tumors. And there was improvement in AIDS human patients, and also autoimmune conditions in human patients like colitis, multiple sclerosis, the neurology problems of type 2 diabetics, and all bacterial and parasitic infections that it was tested against. And at the bottom it says Patent Office versus the FDA. The FDA requires very detailed randomized controls, blinded investigations where the patients as well as the healthcare people do not know who's getting the placebo and who's getting the test agent. Whereas in the patent office, all you have to do is have one very well documented case, which was done primarily in my laboratory at Dallas-Fort Worth Medical Center, because we could easily document, and there were many more different cases. I think there were nearly 60 or 70 in the formal papers that were filed with the patent office where there was benefit. Well, Brooke Army Medical Center at Fort Sam Houston in San Antonio, Texas, is an internationally recognized burn center. And all the military services send their burn patients there. And of course, particularly what they got were flash burns from GIs, from improvised explosive devices, which were very debilitating. And I got a phone call from the burn center by a physician on the staff, and that one of the distributors of Manitech Incorporated that made a topical wound dressing had sent a whole case to the burn center to try in these GIs. And the phone call was that the GIs liked this so much, this aloe-based gel, that they refused to use the FDA-approved Silverdene salve, which was a conventional therapy that you put on wounds, because it burned and it was real messy, turned things black. And they, number one, it reduced the pain that the patients had of burns. They had more rapid healing without scarring, and they cultured the eschar of the edges of the burns, and they found out that the bacteria were not present or killed. It seemed to activate the white cell or body reactions against infection. And at this point, about the rapid healing without scarring and preventing infections were called the guinea pig study. It was done in Costa Rica. This was repeated and seen in these GIs. And in this burn, also often a real problem is when the hands and fingers are burned, and they have to put pressure bandages that look like gloves that put pressure on to keep from scarring, and that you can't even bend at the joints because of excess scar formation, which is called keloids, that was reduced. And they didn't even have to put these pressure bandages on the hands and fingers when they used this man-made aloe gel that contained the acemannan. And of course, it was not messy and it didn't cause problems. Well, the request was to send to the wound center all the data we have from basic science and human data that I've shown you, the titles, and just introduced you that the study was done, but they wanted the complete studies, and they requested that there be an on-site, now it should be SITE instead of on-site lecture. Well, after doing all this, there was silence. And despite having the name and the phone numbers and trying to call the burn center, got no response after sending all this information and having requests for a center. It took over a year to find out what had happened. It happened to be a nurse in San Antonio that was a civilian that had worked in the burn center, went out to find out what happened. Well, all the physicians that were present and saw all the benefits from this natural product of the aloe gel had been shipped out, and some had been sent to hospitals in Germany and Japan and other countries. And there was no medical people present in the burn center that had observed the GI favorable responses to it. And so there was no interest because they had used up all the Acemannan wound gel. And I could probably write a book, but I would hesitate to do it while alive, about all the things that I've run up against, because this is disruptive technology. It challenges conventional beliefs in nothing but FDA-approved drugs. And as I point out, twice I was asked by a company in London to go to Belgium, where they were developing the new laws for the European Common Market for drugs and dietary supplements. And I pointed out that all your man-made organic chemistry drugs interfere with the engineering and design of life that is coded in the genes to suppress symptoms and signs of deviations from normal. And that if you quit taking the drug, which is an ideal drug, those symptoms will come back. That's good business. However, this supplying nutritionally what the genes require to make bioactive molecules to carry out the normal healing and care of wounds and burns, the acemannan, or the aloe molecule, is utilized in the endoplasmic reticulum to make fibroblasts and the types of cells that accelerate the healing and also suppress the burning discomfort. And I suspect that the detail men there in San Antonio that sell Silverdene pulled the strings within the federal government to get all these doctors sent away so that they could go back to utilizing only the FDA-approved Silverdene. And an detailed experience that happened in London in 3 hospitals really amplify and convince me that this is probably what happened, because it happened in 3 hospitals in London, and it was found out in detail that this is what happened. Well, that completes my ability, to the best of my ability, to show you that not only animal experiments, that their biochemistry and mechanisms for handling wounds and burns are similar to humans, and that what you can see in animal models can be translated and obtained in humans when you progress up the process that the FDA has put together, that you progress from tissue culture to small animals to bigger animals and ultimately to humans, that what you find in the animals can be repeated in the humans, and that modern science is validating ancient insight and wisdom and empiric observations that were made before our calendar even started. So with that, we will discontinue until we have our next session together.